today's lecture, we'll consider the problem of measuring drug absorption in translational medicine. There are a variety of methods available to us to measure drug absorption. The most direct is to simply administer test doses of the drug to human volunteers, collect blood samples over time, and measure the drug levels. The problem with this, even though it's very direct, is there can be ethical considerations and obstacles involved if we're talking about a poorly characterized experimental drug, especially if the toxicity is not well known. An alternative is to administer test doses to laboratory animals, rats, dogs, and monkeys, for example. The problem with this is that these animals are obviously not human, and you tend to need to measure rats and dogs and monkeys in order to accurately predict what the human drug absorption is going to be. This is very wasteful of drug material and very wasteful of animals, so there's some moral considerations in terms of animal welfare. So a very useful alternative is in the laboratory to use human cell cultures to measure permeability and use permeability as a predictor for human oral drug absorption. So the objectives of today's lectures, the points that I would like you to take away from today's lectures, are that you can identify the biochemical and physiological processes involved in drug absorption, that you can describe the characteristics of CACO2 cells as a model of human oral drug absorption, that you can take experimental data and compute drug permeability using the fixed law of diffusion, and then you can compare and predict relative drug absorption of one drug to another by comparing what's known as the apparent permeability coefficient values. Why is knowing drug absorption so important? Well, in order to achieve effective drug therapy, you have to have a sufficient fraction of the oral drug dose absorbed. This term is often known as bioavailability. So the more drug that's absorbed, the better the bioavailability, the easier it is to achieve safe and effective blood concentrations of the drug. And this can be best seen in this graph, where we're plotting plasma concentration of a drug versus time. So the drug is administered, it goes through an early absorption phase, where more and more of the drug is absorbed, it appears in the plasma, until at a certain time a peak concentration is reached. And ideally that peak concentration is within the therapeutic range, but below the toxic range. And then the drug concentrations begin to fall off. This is the elimination phase, where the drug is removed from the body by metabolism or excretion, for example. So here we see that oral drug absorption drives bioavailability, and that's what drives drug effectiveness. So it's very, very important to know in advance what the drug absorption of a new compound is going to be. And there are various methods we can do that. We can start out very simply by looking at some physicochemical parameters. So things like molecular weight, hydrogen bonding, lipophilicity, or log P can be measured and calculated. We can start looking at more complicated chemical and biological parameters like solubility and permeability. And permeability is very, very relevant in terms of physiology. And there are actually 5 different processes involved in permeability. First is passive diffusion, represented here in figure A. So here we've got a monolayer of cells, and this is we'll say the intestinal surface, this is the vascular surface. So the drug is administered, and by passive diffusion, the drug moves down its concentration gradient passively, just diffuses through the cell, and appears in the bloodstream. Paracellular transport is somewhat similar. We've got passive diffusion, but in this case, the drug is moving through these gaps or tight junctions that exist between the cells. So it is passing in between the cells rather than through the cells. We also have active transport. This is an energy-dependent process using membrane-bound proteins, and these proteins actively transport the drug against its concentration gradient across the cell. We also have pinocytosis, less relevant to drug absorption, but this involves the drug getting encapsulated in these small bubbles that diffuse through the cell and release the drug on the other side. Lastly, we have efflux pump transport. This is again an energy-dependent active transport using membrane-bound proteins, but in this case the proteins are pumping the drug out of the cell as fast as or faster than it tries to enter the cell. So these are the 5 different processes that can be used in drug absorption and permeability. So experimentally in the laboratory, there are different models we can use to measure permeability, ranging from the very simple and easy to use to the more complicated models. The simplest are to use things like liposomes, or even simpler micelles. So micelles are just a little droplet of lipid. A liposome is actually a sphere of lipid, where you've got this lipid bilayer arranged in a sphere, kind of like a bubble, and then you can actually measure the passive diffusion of the drugs from outside the sphere to the inside of the sphere. You can also use artificial lipid bilayer membranes, indicated in this diagram, and you can measure the transport through active diffusion across one side of the bilayer to the other. Now, the important thing to keep in mind is the absorption process that is mainly being used in these simpler models, the liposomes and the bilayers, is passive diffusion. The more physiological relevant model would include all of those mechanisms that I mentioned in the earlier slide: passive, paracellular, active transport, active efflux, pinocytosis. And that can be best done in using cells, such as these CACO2 cells. And these are actually cultured human colon cancer cells. So the relevance is really there. We're talking about human tissue taken from the intestine, very, very relevant for measuring human oral drug absorption. And this is what the cells look like in culture. They're grown up in these 2-compartment multi-well plates in a monolayer. And this is what the cell monolayers actually look like. Here's a transsection using an electron micrograph, and you can see one surface. You see these finger-like projections. This is known as the apical side, and this arrow is pointing to a tight junction between the cells. Here is a vertical view from the top using immunocytochemically stained cells. And so here's the actual cells, and this lighter green area are the tight junctions between the cells. Here's a cross-section, again using immunocytochemical staining, and we see the apical surface, the finger-like projections here, and the other surface, the basal surface, is right down along here, very simple. Here's a schematic view of these cells. So again, we've got 2 cells, tight junction in between, the apical side with the finger-like projections, the basolateral side right down here. And it turns out that the CACO2 cell monolayer is a very complete model for studying drug disposition. You can measure passive diffusion through the cells, paracellular transport between the cells. There are multiple active uptake transporter proteins on the apical and basolateral surfaces. There are multiple active efflux transporter proteins on the apical and basolateral surface. And in addition, there are multiple drug metabolizing enzymes in the endoplasmic reticulum within these cells. So the experimental and clinical significance of this is not only can these cells be used to measure drug absorption, but they can also be used to get a more complete picture of the pharmacokinetics as well, the absorption and the elimination. And in cases where you've got multiple drugs being given to a single patient at the same time, and the drugs of one drug can interfere with another drug, the so-called drug-drug interactions, because you have so many different mechanisms in place here, these CACO2 cells are also a very valuable model for predicting drug-drug interactions. And it turns out that cancer cells overexpress these efflux transporter proteins. And in fact, that's how these proteins were first discovered, when scientists and clinicians realized that certain cancers were resistant to certain chemotherapeutic agents because they were getting pumped out of the cells faster than they could get absorbed by these transporters. And so this Keiko-2 cell model can be a useful predictor of cancer chemotherapeutic drug resistance. Now, how do we actually use these cells in the laboratories? So here's a cross-sectional diagram of those transwell plates. We've got the larger outer well, the inner insert well, the Keiko-2 cell monolayer growing here, being supported by a polycarbonate filter. So here's the apical surface with the finger-like projections, and this is called the apical compartment, and this is the basal compartment right here. And so what would happen then is the drug being examined is administered here, transported across the cell monolayer, and accumulates in the bottom basal compartment. And what we then do is measure accumulation of the drug over time in the basal compartment, and these data can be graphed out here, where we're looking at accumulation of the drug versus time, and we get this nice linear relationship. Then these data can be plugged into the equation for a fixed law of diffusion right here, and we can calculate a apparent permeability coefficient that's unique for each drug and describes its permeability. And so this is using the concentration of the drug added to the donor compartment, the accumulation of the drug in the receiver compartment, the volume of the receiver compartment, the change in time, the area of the filter support. And so as more and more drug gets transported and accumulated, this value goes up, which increases this value. So what does drug transport then and the P-apt value tell you about drug absorption? Yes, that's right. So the more and more drug that gets transported and absorbed, the greater the P-apt value is. And so in fact, that is what's used experimentally to predict drug absorption. And so here's some data. Here we've got the Keiko-2 cell monolayer, all the different transport processes involved. If we plot the permeability coefficient in Keiko-2 cells versus the percent fraction absorbed with an oral dose in humans, we see that the most highly absorbed drugs have the highest permeability coefficients. The most poorly absorbed drugs have the lowest permeability coefficients, and the ones in between fill out in here. So we get this sigmoidal relationship, but one that is very predictive, using permeability coefficient to estimate human drug absorption. So to summarize this lecture, what we've learned is that drug effectiveness is dependent upon drug absorption, and that Keiko-2 cells are an excellent model for human drug absorption and can be very predictive in the laboratory for human drug absorption. And the important points I want you to come away with are to be able to identify the biochemical and physiological processes involved in drug absorption, to describe the characteristics that make the Keiko-2 cell model valuable for predicting human oral drug absorption, to be able to take experimental data and compute drug permeability using the fixed law of diffusion, and then lastly, to be able to compare permeability coefficient values and then predict drug absorption of one drug relative to another based on those permeability coefficient values. Any questions?